I'm Justin Hamilton, a researcher at the Australian Centre for Blood Diseases at Monash University in Melbourne, Australia. And I'm the lead author on this paper in Science Translational Medicine entitled Disrupting the Platelet Internal Membrane Impairs Thrombosis Independently of Canonical Platelet Activation. So what is our paper all about? Well, in short, we have identified an approach to prevent the blood clots that cause heart attacks and strokes without preventing the blood clots that stop you from bleeding. How did we do this? Platelets are the small cells in your blood responsible for forming blood clots. And current therapies designed to prevent the blood clots that cause heart attacks and strokes prevent platelets from functioning correctly. But they do this indiscriminately. They stop the function of all cells all of the time. And as a result, you're always walking a fine line between preventing pathological blood clots and causing bleeding. And as a result, we've really hit a ceiling in the efficacy of current therapies. So in this study, we identified an approach with the potential to overcome this major limitation. We developed the first compounds that block the function of a particular enzyme located inside platelets that we have identified is really only important in the setting of pathological blood clot formation, such as those that underlie heart attacks and strokes. And so using this approach, we don't get an overall block of platelet function all of the time, only when you need it. And so what is the major advance of our study? Well, we think that the major breakthrough of our study is that our approach is different to every other drug that's used today, in that the compounds that we have developed have no effect on platelets until they're called into action to form a pathological thrombosis. So the cells behave entirely normally in every aspect that we have examined in the laboratory, except when they are triggered to form a blood clot under pathological conditions, such as those that would trigger a heart attack or a stroke. And so we think the targeted nature of this approach is likely to substantially reduce the bleeding side effects observed with current therapies. What is the next step in our research? Well, our study has made the first inhibitors of the target enzyme inside these platelets, and we showed that these first compounds are very effective antithrombotics in human blood and in animal models. But the compounds we developed are not yet drugs. And so the first aspect of our research is to improve on the compounds that we have in hand in order to develop candidates that are likely to be used as drugs in human clinical trials. The second aspect is to identify the patients most likely to receive benefit from the approach that we have developed here. In the first instance, we are looking at patients that are at some of the highest risk of thrombotic complications. For example, those with underlying diabetes or with pronounced hyperlipidemia, high cholesterol.